Let's examine this annotation on Arabidopsis to explore some additional functionality in its annotation details dialog. First, let's take a look at the Metrics tab. In this view, we can see the annotation with its exons numbered at the top. While the exons are always drawn to scale, some introns may be abbreviated to save space, as indicated by this glyph. Click anywhere in this view to move the hairline cursor to the clicked position. The readout on the right side of the hairline displays the exact cursor coordinates as measured from the start of the annotation, the start of the current exon, and so on. The readout at the bottom always displays the global position of the cursor, as well as its local position relative to the start of the annotation. If the cursor is inside an exon, then this readout will also show its exon number, exon length, and offset modulo 3. You can press the left or right arrow keys on the keyboard to move the cursor by exactly one nucleotide. Hold down the Control key and press the arrows to jump directly between exon and CDS boundaries. Drag the mouse to select a fragment of the annotation sequence. The mouse will snap to exon and CDS boundaries, and the readouts will update to display the length of the current selection in global as well as local coordinates. You can hold down the Shift key and press the left or right arrow keys to change the selection one nucleotide at a time, or hold down both the Control and Shift keys to quickly expand the selection to the next exon or CDS boundary. You can also double-click an exon to select it. The selection in the Metrics view is synchronized across all of the sequence tabs for this annotation. For example, the sequence of the exon we've just selected is also selected in the Protein tab, the Splice Sequence tab, the Translation tab, and the Unspliced Sequence tab. And if we make a selection in the sequence text, it will be reflected in the Metrics view as well. The gene models in the Metrics view are always shown in the forward orientation with the 5' end on the left. This means that if we open the Metrics view for a model on the reverse strand, then the cursor coordinates will decrease from left to right. Now, let's take a look at the Transcripts tab. This view displays all the splice variants for the current gene, lined up for comparison. The current transcript is marked with a darker background and this triangle icon on the side. The names of the transcripts are abbreviated to save space, but you can mouse over any transcript to display its full name and key metrics in a tooltip. The exons of all other transcripts are drawn with a darker color if they agree with the current splice variant, and in a lighter color otherwise. The numeric position labels at the top of the screen list all of the exon and CDS coordinates across all of the splice variants in the locus. As you move the mouse over the transcripts, only the position labels relevant to the sequence fragments under the cursor are revealed. These fragments are also highlighted in yellow. As you can see, in this case, there was not enough room to draw all the positions on the screen, but we can always zoom in by rolling the mouse wheel to see more details. As usual, we can zoom in even further to see the protein translations. We can also customize the view by using the controls at the bottom of the screen. For example, we could hide the position labels completely, or reveal only the ones that disagree with the current transcript. In addition, this checkbox trims the common digits at the front of every position, and this checkbox displays each position modulo 3. It is also possible to hide the transcript names, or to show them in full in the middle of the screen. By default, these transcripts are ordered by name. However, we can also sort them by unspliced sequence length, with longer transcripts on top. Or we could instead sort them by CDS length. In this case, this additional option will group together all the transcripts with the same CDS structure, ignoring UTRs. However, you can also manually expand or collapse each group by using these buttons on the side. Finally, you can click a splice variant to open its own transcript view in a separate dialog in order to compare the two models side by side.